All right, let's dive into some really interesting science today. We're tackling a challenge that affects millions: neurodegenerative diseases.
Yeah, things like Alzheimer's, CTE, conditions linked to this protein called tau.
Exactly. These are diseases where basically the tau proteins kind of go rogue.
Right. They stop doing their normal job,
Yeah.
and instead they misfold. They clump together into these harmful little threads or fibrils in the brain.
And, for researchers, one of the biggest hurdles has been figuring out how this misfolding even starts.
How it spreads.
And honestly, how to detect these diseases reliably before they cause irreversible damage.
Yeah, because right now, diagnosis is often based on, well, symptoms, surveys.
Yeah.
Not direct biological evidence from the brain while someone's living.
It's a huge bottleneck, a real problem.
Absolutely. We really need better tools, better models to understand these, really complex processes at a fundamental level.
Which brings us neatly to the core of this deep dive today. Researchers at Northwestern and UC Santa Barbara. They've just announced what feels like a pretty major step forward.
Yes, they've managed to engineer the first synthetic piece of the tau protein, a fragment that actually behaves like a prion in the lab.
A synthetic mini prion? Is that what they're calling it?
That's the term, yeah. It's a breakthrough tool, potentially.
Okay. So our mission here is to unpack what this synthetic mini prion actually is, how they made it, and what surprising things it revealed about tau misfolding.
And trust me, one factor is something you probably wouldn't guess right away.
Right. And we want to explore why this could be such a game changer for, finding new ways to diagnose and maybe even treat these conditions.
Okay, but before we jump right into the new stuff, maybe we should just quickly recap why studying tau misfolding is so tricky in the first place.
Good point. So you have your normal, healthy tau proteins in your brain cells doing their jobs.
Stabilizing structures, things like that.
But in these diseases, if a normal tau protein bumps into one of these misfolded tau fibrils...
It gets templated.
Templated. Explain that a bit more.
Well, the normal protein changes its shape. It adopts the same misfolded shape as the fibril it encountered.
Ah, so it conforms to the bad shape.
Exactly. And then that newly misfolded protein can go on and template other normal proteins.
Okay, so it's like this toxic chain reaction spreading through the brain tissue.
Right. And that ability to template, to propagate the misfolding shape, that's what we mean by prion-like behavior in this context.
And we should probably be super clear here. When we say prion-like, we're talking about the self-propagating misfolding within the brain's tau proteins.
Yes. Very important distinction. We are not talking about the kind of infectious person-to-person spread you see with classic prion diseases like CJD.
Got it. So it's the mechanism of templating within the brain tissue that's the key similarity here.
Precisely. Now, scientists can get a look at the structure of these harmful tau fibrils. They use amazing techniques like cryo-electron microscopy, cryo-EM.
Which gives these incredibly detailed pictures.
It does. But the catch is the samples they use. They typically come from brain tissue collected after a patient has passed away.
Which, you can imagine, makes it incredibly difficult to study the very beginning of the misfolding process.
Or to experiment easily.
Yeah.
You can't readily test drugs or different conditions on those samples in a dynamic way.
It's like trying to figure out how a complex machine broke down by only looking at the wreckage afterwards.
Exactly. You're missing the crucial early steps. So these researchers, they had a really smart, sort of different approach.
Okay.
Instead of wrestling with the whole very long, very complex, full-length tau protein, which is hard to make perfectly in the lab and hard to control,
right,
they asked, what's the absolute shortest possible piece of the tau protein that still does the bad stuff, that still misfolds and forms these disease-like fibrils?
Like finding the minimal essential component for the trouble.
Precisely that. And they managed to zero in on a specific, very short segment, just 19 amino acids long.
Wow, only 19. Out of how many in the full protein? Hundreds.
Yeah, the main forms have hundreds, so 19 is tiny. They called this fragment JR2R3.
JR2R3.
Okay. And crucially, they designed this fragment to include a specific mutation, P301L. It's a mutation often found in patients with certain inherited forms of tauopathy.
Ah, okay. So they didn't just pick a random snippet. They chose a region implicated in disease and deliberately included that known disease-linked mutation. Smart.
Very smart. And here's the real breakthrough. They found that this tiny 19 amino acid peptide with that P301L mutation could do two absolutely critical things.
What were they?
One, it could form those harmful fibrils by itself. It could act as the initial seed.
Okay, so it could start the process.
Right. And two, maybe even more remarkably, it could take full-length, normal tau proteins and template their misfolding and aggregation.
Wait, hold on. This tiny little 19-unit piece can actually convince the big full-length protein to misfold and clump up.
That's what they found. It seems almost unbelievable, but yes.
That's kind of amazing.
It really is. One of the lead researchers, Song-Gi Han, described it perfectly. She said they made a mini version that is easier to control, but it does all the same things that the full-length version does.
Wow. So it acts as that seed, and it causes the normal full-length tau to misfold and join the growing fibril clumps.
Exactly.
Okay, that is powerful, because now you have a much simpler system, right? Easier to handle in the lab, but it still captures that absolutely essential disease process, the templating of the normal protein.
Precisely. And they didn't just stop there. They used cryo-EM to look at the actual structure of the fibrils formed by their synthetic mini-tau.
And what did they see?
They confirmed that the presence of that P301L mutation in their midi-peptide actually directed the misfolding into a very specific structure, a specific shape, that is often seen in fibrils taken from actual patient brain samples with that mutation.
So it's not just misfolding randomly, it's misfolding in a disease-relevant way directed by that mutation.
Yes. It strongly suggests that mutation is like a key, turning the protein towards this specific problematic structure.
Okay, this is fascinating, but it brings up a puzzle, doesn't it?
What's that?
How do these protein segments, which start off all floppy and disordered, how on earth do they manage to arrange themselves into these incredibly neat, ordered stacks that make up the fibrils?
Yeah, that's always been a bit of a head-scratcher.
It feels like, I don't know, throwing a handful of wet noodles onto a plate and expecting them to land in a perfect straight pile. It just seems counterintuitive.
It does seem counterintuitive. And as the researchers themselves noted, it really doesn't make sense thermodynamically unless there's some other factor helping out.
Okay, and this is where things get really interesting. You mentioned something unexpected.
Yes. This is the surprising part. They uncovered a crucial player that had been largely overlooked before: the immediate environment around the protein.
The environment, like other molecules.
Specifically, water. Simple H2O.
Water? How can water be playing such a direct role?
Well, what they discovered is quite subtle. That P301L mutation, it doesn't just tweak the shape of the tau peptide itself.
Okay.
It also slightly changes the structure and the behavior of the water molecules right there, immediately surrounding that specific mutated part of the peptide.
Hold on. Water having structure. I mean, it's a liquid, right? Not ice.
Right, not like a rigid crystalline.
Yeah.
But water molecules are polar. They have positive and negative ends. And when they're near surfaces, like the surface of a protein, they don't just tumble randomly like they do in bulk water.
They can arrange themselves a bit more.
They can become slightly more ordered. And it seems this P3OML mutation encourages a more structured arrangement of water right around that mutation site.
Okay. So you have this little patch of more organized water molecules near the mutation.
Exactly. And this structured water then influences how different peptide segments interact with each other.
Oh.
It appears to essentially pin them together temporarily.
Wow. So the water molecules influenced by that disease mutation are acting almost like the tiny little clamps or guides.
That's a good way to think about it. They hold these floppy, partially misfolded protein strands in the right position, just close enough and in the right orientation for long enough that they can then click together properly into that very stable ordered stack, the fibril.
And that stable fibril stack, once formed, is the thing that then goes on to template more normal tau protein.
Exactly. That's the template that uses its prion-like behavior to recruit and misfold other tau proteins, perpetuating the whole cycle. The water helps build the initial template.
That's really quite a revelation, the role of water there.
It really adds a new dimension to how we think about these folding processes.
So, okay, let's step back. What does this whole discovery, the synthetic mini-tau, the crucial role of water, what does it all mean for the bigger picture, for fighting these diseases?
Well, it means this synthetic model is potentially an incredibly powerful new tool because it's simplified, because it's controllable. Scientists can now use it to really precisely study those fundamental interactions, the protein-protein interactions, and that previously hidden critical role of the surrounding water, all driven by that specific mutation.
And if you can really zoom in on the exact molecular players and mechanisms, like how that specific water structure helps pin things together,
then you can start thinking much more precisely about how to stop it, how to interfere with that process.
Right. Makes sense.
So, by being able to create and replicate these specific disease-relevant structures in a controlled lab setting, this research really gives us a direct target.
A target for what? Drugs? Diagnostics?
Both, potentially.
Yeah.
The researchers emphasize that once these tau fibrils form, they're incredibly stable and just keep recruiting more normal tau, seemingly forever.
So you have to stop them forming or stop the recruitment?
Exactly. The key therapeutic goal becomes figuring out how to block this activity. And this synthetic mini-cow model, it provides a fantastic platform to test potential drugs or other agents.
Agents specifically designed to disrupt that templating process, maybe even targeting those water interactions.
Perhaps. Or blocking the sites where normal tau binds. It opens up new possibilities. And yes, it could also help develop better diagnostic tools.
Like the biomarkers we mentioned earlier, things that could spot these specific misfolded structures much, much earlier in the disease process.
That's the hope. If you have a reliable way to make the specific pathogenic structure in the lab, you might be able to develop probes or antibodies that specifically recognize it.
Okay, this really does sound like it could shift things. Moving from just observing the damage late in the game to understanding and maybe intervening in the root cause much earlier.
That's the potential, yes.
Yeah.
It's a fundamental step forward in our ability to model and probe the disease mechanism.
All right, that was definitely a deep dive. So just to quickly recap the key points for you, our listener, we started with the huge challenge of tauopathies, diseases like Alzheimer's, where tau proteins misfold and clump together.
Then we talked about the breakthrough. Scientists creating this controllable synthetic mini prion, just a tiny 19 amino acid piece of tau with a specific disease-linked mutation.
A piece that could still trigger the misfolding of the full-length protein.
Right, and then came that really surprising twist, the crucial role of seemingly simple water molecules, structured by that mutation, in essentially pinning the misfolding proteins together, helping them form those toxic fibrils.
And the big picture is that this research gives scientists a powerful new lens, a new tool to understand the nitty-gritty mechanics of these devastating diseases.
And hopefully that provides a clearer path forward for developing future diagnostics and, crucially, new therapies.
Absolutely. And, thinking about that unexpected role for water as something so basic, so everywhere in our bodies, playing such a fundamental structural role in a complex disease process like protein misfolding. It really makes you wonder, doesn't it, what other sort of mundane environmental factors, things maybe we just take for granted inside our cells, what else might be quietly influencing disease mechanisms in ways we haven't even begun to uncover yet?
That's a really interesting thought. It definitely raises fascinating questions about the whole context, the cellular environment, and how it shapes these critical biological events.
Something to ponder, for sure.